So today we'll be continuing with the topic of molecular inheritance. So we had stopped the unit at replication, so that is where we'll begin the topic. In the previous series, we had learned about central dogma. DNA exists in a double helix conformation. So in a given DNA segment, we have two strands. Now these two strands we had learned are antiparallel, meaning they have opposite polarity and they are complementary to each other. So when Watson and Crick they proposed the model of DNA, double helix model, immediately by seeing this structure, they also realized how DNA could replicate, and they gave the semi-conservative model of DNA replication. Semi-conservative model of DNA replication. This was given by Watson and Crick. Okay, so what does semi-conservative model tells us? Semi-conservative model tells us that each strand, now DNA has two strands in antipolarity conformation. Now both of these strands, both of these parental strands are going to act as the template. They both are going to act as this template and they will be coding for a daughter strand. Now the daughter strand that will be formed will be in opposite polarity or will be antiparallel to the parent DNA. And at the end result of replication, what we get? We get a DNA strand that has one parental template strand and one daughter strand. So here there is a parental strand and one daughter strand. So this particular model is known as semi-conservative model because in each round of DNA replication, one parental strand is conserved along with the daughter strand. Thus, this is known as semi-conservative model. The experimental proof of this was given by Messelson and Stahl experiment. Messelson and Stahl experiment was able to prove that DNA replication followed semi-conservative model. Okay, it followed semi-conservative model. So what they did is they took some E. coli culture and they grew it in N15 medium. N15 is a heavier isotope of nitrogen and they grew E. coli bacteria in N15 medium for some generations. After some generations, what they did, they transferred it in another medium that was N14. N14 is a lighter isotope of nitrogen. So first, when they were growing the bacteria in N15, they took a sample of it and centrifuged it. In the process of centrifugation, the mixture is blended, and as it is blended, the mixtures separate according to their density component. Okay, so N15 being heavier settled and formed a band at the bottom. Now what we need to know is when we are providing N15 in the medium, basically, the bacteria will utilize N15 and incorporate them into their DNA. We studied that in the structure of DNA, we have nitrogenous bases, right? So for synthesizing DNA, the bacteria is going to utilize the N15 that is provided in the medium to make their DNA. So that is why we are getting an N15 band here. Now, after the bacteria had been transferred, the first generation that we got was again centrifuged. And on centrifugation, what did we observe? We saw that this time we got a band that was lighter than the N15 band. So what is happening? Why are we getting two bands till now? So first let's see what is happening in the molecular basis. So when the bacteria was originally in N15 medium, it incorporated N15 into its DNA. So as a result, the strands that were being formed were all N15 strands. They all had N15 in their DNA. After they had been transferred and the first culture was taken, now these two strands in the first generation, they are going to separate and they are going to both act as the template. And the new daughter strand that will be synthesized will only use the material that is there in their surrounding. So in their surrounding now they only have N14. As a result, the new daughter strands that will be formed will only use N14, right? So they will be lighter. So here we can see we are getting hybrid. We are getting hybrid. N15, N14 hybrid, right? We are getting a hybrid. Now, due to the presence of this hybrid, we get a band that is intermediate and is lighter to N15. Okay. And finally, what they did was, after few more generations, but in the same medium, in the same N14 medium, but after some generations, they again took a sample. And this time, what did they get? They got two bands. They got a hybrid band that was N14, N15 band, and then they got a new lighter band N14. So what might have happened? Again, when this bacteria is going to replicate itself, let's just concentrate on this part. When it will replicate itself, now both of them are going to act as the template. Now this strand will form again a new daughter strand. The daughter strand is going to use N14. So both of these strands are N14, right? As a result, we will get a new strand that is completely composed of N14 strands, right? And similarly, this can also act as the template. And here we are going to get a hybrid. Okay. So that's the result. At the end, we were getting two bands. Okay. A heavier band and a lighter band. So this experiment proved the fact that DNA was semi-conservative. It was semi-conservative replication that DNA was following, right? Okay. So for DNA replication, what are the prerequisites or what are the materials that we require? We of course require a template strand. And then we require an enzyme, a key enzyme that could polymerize and form the daughter strand. The key enzyme in this case is DNA polymerase. Now DNA polymerase, in case of prokaryotes, are of three different types. We have DNA polymerase 1, 2 and 3. Okay. So in case of prokaryotes, we have three different DNA polymerase, and out of these three different polymerase, DNA polymerase 3 is the major enzyme that is involved in polymerization and formation of the daughter strand. In case of eukaryotes, we have around 14 DNA polymerases. So we have a large variety of DNA polymerases when it comes to eukaryotes, but we only have to learn three important ones. We have DNA polymerase alpha, DNA polymerase delta, and DNA polymerase epsilon. Okay. So alpha is going to help me initiate the process of DNA replication. So we require an enzyme, DNA polymerase, right? And this DNA polymerase is DNA dependent DNA polymerase, right? Why? Because it is dependent on the template strand. Okay. So besides this, we need the substrate. So if I have to synthesize a new daughter strand, for making the new daughter strand, I need nucleotides, right? DNA is composed of nucleotides, so I'll require nucleotides as the substrate. So in the case of DNA replication, the substrate is a bit different. Instead of using just nucleotides, we use nucleoside triphosphates. Nucleotide triphosphates. Okay. Now, as the name indicates, they have 3 phosphates. Right. So nucleoside triphosphates will have 3 phosphates. And what is the advantage of possessing these 3 phosphates? They can break this phosphate bond and release energy. And this energy can be used in production of the daughter strand. Okay, so DNA replication is a process that requires energy. So where is the energy coming from? It is coming from the substrate itself, that is your nucleotide triphosphate. Okay. Now in DNA replication, we have to remember the structure of DNA. The DNA is in double helix form. It is in double helix form. For the 2 strands to be replicated, first we need to separate the 2 strands. But we cannot separate them all together, as it would be a very much energy intensive process. Rather, what happens is DNA opens at small regions, right? DNA opens at small regions rather than opening all together to conserve energy. At the site where DNA opens, that site is known as ORI, or that is origin of replication, ORI site. Okay, so it is at the ORI site that the hydrogen bonds are broken and DNA is opened, right? We know that the double helix structure conformation is maintained with the help of hydrogen bonds or base pairing of nucleotides, right? So if I have to break that, I will need some sort of enzyme that is going to break the hydrogen bonds and help me separate the 2 strands. So here we require an enzyme helicase. Helicase helps me to unwind the DNA, basically to open the DNA, right? So DNA helicase is going to help me to open or unwind the DNA at the ORI site. Okay. Now at the ORI site, you basically have to open the DNA. So ORI site is rich in 80 base pairs, right? Why? Because 80 base pairs are more easier to break than GC. 80 only has 2 hydrogen bonds, whereas GC has 3 hydrogen bonds, right? So as a result, the ORI site is 80 rich. It has major of 80 nucleotides, so that it's easier to break. Now besides the helicase, we have single strand binding proteins. The DNA is existing in a stable conformation. From there, you are breaking apart the DNA. You are separating the 2 strands of DNA. So then to stabilize the DNA again, we will require some particular enzymes known as single-stranded binding proteins. Okay, they bind to single strands, as their name indicates, and help to stabilize them. Now besides this, as DNA is opening up, it's going to generate tension ahead of the replication fork. So we require another enzyme, topoisomerase, to remove the tension. So these are some of the key enzymes that we would require. This is the ORI site. At the ORI site where DNA is opened, there is formation of a replication fork. A replication fork is formed here. You can see a Y-shaped replication fork is formed where DNA has opened. This is known as replication fork. And this whole bubble that is formed is known as replication bubble. Replication bubble. So these were some of the enzymes that we required, and we have seen what all do we require. So let's start the process of DNA replication. So for DNA replication to start, we need an ORI site from where helicase will start to open them, right? And what we see is, this is the replication fork. We have two strands, 5' to 3', and another one is 3' to 5', right? So we know that DNA polymerase can only add new nucleotides to the 3' OH side of an existing strand. So DNA polymerase requires an existing strand, and in that existing strand it can add nucleotides only to the 3' end. We know that DNA has a 5' end and a 3' end. So DNA polymerase can add new nucleotides only to the 3' end. So 3' end is known as the growing end. So DNA replication will always proceed from 5' to 3', right? But due to this particular nature of DNA polymerase, there is an additional requirement. What did I tell you? The DNA polymerase requires an existing chain. Existing chain, right? To an existing chain can only DNA polymerase add new nucleotides, right? Due to this, we also require a primer. What is a primer? A primer is a single-stranded stretch of RNA nucleotides, right? It is a short stretch of RNA nucleotides, right? And this short stretch of RNA is made by primase enzyme, okay? So we require a primer to which DNA polymerase can add new nucleotides and begin the synthesis of daughter chain. So if you see here, this chain is 5 to 3, right? This is 3 to 5. I told you replication will always proceed in 5 to 3 direction. So here in this direction, DNA replication can very easily proceed, right? You will just require a primer here. And to this primer, DNA polymerase will come, act on it, and add new nucleotides depending on the complementarity. Okay? So new nucleotides are added based on the complementary base pairing. Okay? So here the strand synthesis will be continuous. Here the strand will be synthesized in a continuous fashion, and this strand is going to be named as the leading strand. Leading strand. Whereas in the template strand that is going from 5' to 3' direction, there how will the strand be produced? It will be produced in the opposite direction, right? 3' to 5'. But we know that DNA polymerase only grows a chain in 5 to 3'. As a result, the synthesis here is a bit different. Here there would be discontinuous synthesis. Okay. In case of leading strand, we require just one primer whereas in lagging strand we require multiple primers. What are these? These are lagging strands, also known as the Okazaki fragment. Okay, Okazaki fragments. So once DNA replication is complete, we have DNA polymerase that is going to polymerize the whole chain. So once the replication is complete, this primer region is removed, and after that, all the nucleotides, all these short stretches, they are joined together by DNA ligase. Okay. So we are almost done with DNA replication. So where does DNA replication takes place? It takes place in the S phase of cell cycle. S phase of cell cycle. Okay, and what is its speed of replication? It is around 2000 base pairs per second. Okay. So with this we have completed replication. We have understood how a DNA strand is going to synthesize another DNA strand. Okay. So the next thing that we need to learn is transcription. Transcription. Okay. Transcription is a part of gene expression. We know that a genetic material is able to express itself, right? A genotype produces a phenotype. So how is that a gene is able to express itself? A particular gene expresses itself with the help of two processes, transcription and translation. In transcription, what is happening? The DNA segment, that is a particular gene, will code for a mRNA. Will code for a mRNA. Okay. So transcription is a process that is very much similar to replication, but what are the differences? In replication, both of these strands are completely opened, and replication occurs in both of these strands from start to end. Transcription only happens for a particular gene segment that is required, right? And in transcription, only one particular strand is going to participate in transcription, unlike replication. Okay. So which strand will be participating? The strand will be participating is 3 to 5 prime, right? Because why? We would get a 5 to 3 prime daughter strand here, or mRNA transcript here, right? So the strand that is going to participate in the formation of an mRNA strand is the strand that has the polarity 3 prime to 5 prime. This is known as the coding strand. This is known as the coding strand, right? Sorry, this is known as the template strand. Okay. So this is known as the template strand. This is the template strand. This would act as a template strand, and a mRNA transcript would be synthesized from this, whereas the other strand is the coding strand. Why we are calling it as the coding strand? Because the mRNA formed would be identical to the coding strand. Its sequence of base pairs would be identical to the coding strand, right? Whereas to the template strand, it would be complementary. Okay, so for example, if your template strand, we have A, T, G, C. So in mRNA, we are going to have opposite of A pairs T, but in mRNA we do not have thymine, instead we have uracil, right? U, T, A, C, G, right? And what would be the sequence here? The sequence here would be exactly same of as the mRNA, except that in presence of uracil, thymine would be there, right? So that's the reason. Okay. So in the process of transcription, what all is required? We again require an enzyme require a template strand Now the gene segment is divided into certain regions for the process of transcription. We have a promoter region. We have a promoter region. Then we have the structural gene. And then we have a terminator. So what is the role of promoter region? Promoter region helps to bind the enzyme involved in transcription. Okay, helps to bind the enzyme and transcription factors that are required for the process of transcription. So what is the enzyme that is required here? We require, now we are producing RNA, right? So it is RNA polymerase. RNA polymerase is a DNA dependent process because it's dependent on DNA to produce the RNA. Okay, so the enzyme required is RNA polymerase, right? So RNA polymerase and transcription factors would bind to the promoter. Okay, as a result, throughout evolution, the promoter sequence has been conserved. Throughout the evolutionary history, it has been conserved. Why? Because if there's a change in promoter, the RNA polymerase will no longer be able to bind to the promoter. It will not be able to be recognized. As a result, then there will be no gene expression. Okay. As a result, the promoter region, like your Tata, -10, -35, all of these regions are conserved. Okay. Then we have the structural gene. Structural gene is the region that would be coded in the mRNA, right? This much region would be coded in the mRNA. It will be coding for the mRNA. Okay. And then we have a terminator. On reaching the terminator, the process of transcription stops. That is why it's known as a terminator. Okay. Now, RNA polymerase, in case of prokaryotes, we just have one RNA polymerase. Okay, one RNA polymerase. And this one RNA polymerase would transcribe the different classes of RNA that are there. That is, we have ribosomal RNA, we have mRNA, and we have tRNA. So this one RNA polymerase, in case of prokaryotes, is going to transcribe all the three different classes of RNA. Okay, whereas in case of prokaryotes, or in case of eukaryotes, we are going to have three different RNA polymerase. We are going to have three different RNA polymerase that is going to transcribe each different class of RNA. So in case of eukaryotes, we have RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. RNA polymerase 1, 2, and 3. RNA polymerase 1 transcribes ribosomal RNA. It transcribes ribosomal RNA except 5S RNA. 5S ribosomal RNA. Okay. So what are ribosomal RNA? Ribosomal RNA are part of ribosomes. They help in formation of ribosomes, right? So ribosomal RNA of different types. Out of those different types, except 5S RNA, the rest of ribosomal RNA is transcribed by RNA polymerase 1. Okay. Okay. And what are these ribosomal RNA? These are 18S, then we have 28S and 5.8S RNA. Okay. These three, in case of eukaryotes, are coded by RNA polymerase 1. RNA polymerase 2 is very important because this is going to code for your mRNA. Okay, this is the one that codes for your mRNA. Okay. So this is the one that is involved in protein formation, right? So this is going to encode for mRNA. RNA polymerase 3 is involved in encoding tRNA and 5S RNA. So what we see is in eukaryotes, there's additional complexity. There is 3 different classes of RNA polymerase, whereas in prokaryotes, there's just one RNA polymerase that is going to transcribe all the different types of RNA that are there. So the key enzyme involved in case of transcription is RNA polymerase. So we have seen promoter region. Promoter region is where RNA polymerase binds to, right? And in the structural gene, the region where transcription starts, where the process of transcription starts, or where the formation of coding of mRNA starts, is known as the start site. Okay, and it's denoted by +1. Okay. +1 denotes the start site where the actual process of coding for mRNA starts. Okay. The site before +1 are referred to as upstream sequences. Any sequence that is present before the start site or preceding it is known as the upstream sequences. And sequences that come after it are known as downstream sequences. All the upstream sequences are denoted with a minus sign and the downstream sequences are denoted with a plus sign. Okay. As an example, we can say promoter is present upstream to that of the structural gene, or the terminator sequence is downstream to that of structural gene. Okay. So how is the process of transcription started? In case of prokaryotes, this occurs in the cytoplasm itself, right? Why? Because they do not have a separate nucleus region, whereas in case of eukaryotes, the process of transcription occurs in case of nucleus, right? In nucleus. So at the promoter site, first of all, for a process of transcription to start, the RNA polymerase will come. It will recognize the particular sequence that is a promoter and bind to it, right? So in case of prokaryotes, I had told you that they have just one particular RNA polymerase, right? RNA polymerase. Now this particular RNA polymerase has a core enzyme part, and to this they have a sigma factor. So RNA polymerase is made up of core enzyme and sigma factor. Okay. Now this sigma factor is needed for initiation only, right? It is needed for initiation and recognition of the promoter sequence, right? So when RNA polymerase first binds with the promoter region, then it is sigma factor that recognizes the promoter sequence, right? After recognition, the function of sigma factor is done. So what will it do? It will dissociate from the RNA polymerase and only core enzyme will remain, right? And rest of the transcription process is done by the core enzyme. Sigma factor is involved in recognition and initiation of transcription. Okay. So after that, the process of transcription is very much similar to replication. The new strand will be produced, right? Now how does it terminate? In case of prokaryotes, there are two ways in which transcription can terminate. The first method is through the sequences in DNA itself, right? The sequences in DNA itself are going to stop the process of transcription, right? So in that case, what we call them, it will called as the intrinsic mechanism of termination, right? Why? Because the sequences that are present in the terminator itself leads to termination of your transcription, right? So here there is a hairpin loop formation. the mRNA that is transcribed is going to form a hairpin loop, and as a result it is going to arrest the process of transcription, right? So something like this will be formed and it will stop the process of transcription, right? Another way is they will require rho protein. A rho protein will come and bind to the terminator sequence and then will stop the process of transcription, right? So this was all about the process of transcription. Yeah. Next, before moving on to the your particular next process, that is translation, we should know about genetic code. Okay, genetic code, right? So we know that right now we know that the DNA sequence is based on the sequence of nucleotides or sequence of base pairs that are present in DNA, right? So if I have to denote the sequence of a particular region, I will simply write the base sequence that is there in that particular sequence, right? So scientists, they know that there are around 20 amino acids. That are around 20 amino acids, right? We have 20 standard amino acids. And how many bases do we have? We have 4 bases, right? So a physicist, Gamow, was able to hypothesize the fact that if there are 20 amino acids and there are just basically 4 basic bases that are there in our DNA, then a combination of these bases would be responsible for coding of 20 amino acids, right? Why? Because we know that mRNA is going to code for a protein, right? And in mRNA, what is there? There are nucleotides. And these nucleotides are going to code for a amino acid, right? These nucleotides are going to code for amino acids. So what he hypothesized that 4 bases, combination of 4 bases is going to code for 20 amino acids. And he said that 4 raised to the power 3 could give rise to 64 codons, right? So this is a possible confirmation, right? So he said that the codon is triplet, right? It is triplet. So we came to know that the codon is triplet in nature. That means 3 nucleotides together comprise a codon. So codon exists in the group of 3s, right? So when we are reading the genetic code, we often club the genetic code first into codons and then we read them. Now each codon or each triplet is responsible for producing 1 amino acid. It is going to code for 1 amino acid, right? The genetic code is specific. That means one particular codon will always code for a particular amino acid, like your AUG is specific for methionine, right? Okay. And we know our codon is unambiguous. That means there is clarity in that. And another property is our codon is degenerate. Degenerate means multiple codons can code for the same amino acid. Multiple codons can code for the same amino acid, right? So these were some of the properties of the genetic code. Now that brings us to the topic of mutations. We can finally understand why a mutation is happening. We know that mutation occurs due to an alteration in DNA sequence, right? So here I have the reading frame of DNA. The reading frame of DNA is read from the +1 site, right? +1 site. From the +1 site, we start to group them, right? So that is known as the reading frame of DNA, right? So what kind of mutations can occur? sometimes a single base pair, a single base can be substituted, right, by another base. It can be substituted by another base, right? So what is happening here? One single base is substituted by another base. Is the reading frame changed here? Does any change occur to the reading frame? No, right? Even if it gets substituted, the reading frame remains the same. The reading frame is not disrupted, right? So such kind of mutations where just one base pair is mutated is known as single base pair mutations. Single base mutations. Where usually there is substitution of a single base. This is also known as single nucleotide polymorphism. Right? A very good example of this is the disease sickle cell anemia. Sickle cell anemia. In sickle cell anemia, there is a single base substitution of adenine to thymine. Now, due to this single base substitution, the amino acid that it was coding for changes, right, from glutamic acid to valine, leading the person to have sickle cell anemia, right? Okay. Now, what if instead of one base being substituted, one base is deleted? One base is deleted. Then the reading frame gets altered. The reading frame gets altered. If one base is deleted, what would happen? The reading frame would change. The reading frame would change, right? So whenever there's a change happening to the reading frame, right? So such mutations are known as frame shift mutations. Frame shift. Why? Because they're shifting the frame, right? So if there is addition, deletion of one or two nucleotides, that is known as frame shift mutations, right? If three particular nucleotides are added or deleted, then that is not going to change the frame, right? Okay. Now let's talk about translation. Okay. Now in case of prokaryotes, as soon as they finish with the termination, the process of translation starts, right? So in case of prokaryotes, the process of transcription is coupled with translation, right? Transcription is coupled with translation. So both the processes go hand in hand. As the gene is getting transcribed, the translation of that particular mRNA will also start. So both the processes, they are coupled with each other, right? Because in prokaryotes, we see that there is no particular space difference that is happening. Both the processes are occurring in the same space, that is cytoplasm. In case of eukaryotes, what we observe is that both the processes are separated in space and time. Both the processes are separated in space and time. So transcription is going to occur in nucleus. And then the mRNA that is formed is going to get transported into the cytoplasm, where translation would occur. Now, because a different space is required here, the process of transcription and processing here is a bit complicated, right? So the mRNA formed in case of eukaryotes is going to undergo some sort of processing before leaving the nucleus, right? So in the nucleus itself, some processing will be undergone by mRNA, and this processing will help the mRNA to be stabilized even when it is present in the cytoplasm, right? So next we are going to learn about mRNA processing. Now mRNA processing only takes place in case of eukaryotes. This process is not seen in case of prokaryotes. So in mRNA processing, what can we see? There are three processes that are involved in mRNA processing. There is capping, there is tailing, and then there is splicing. Okay. Let me repeat again. All of these three processes are seen only in case of eukaryotes. There's capping, there's tailing, and then there's splicing. Okay. So in capping, what is happening? The hnRNA, now before the mRNA undergoes processing, we call it as heterogeneous RNA. It is known as heterogeneous RNA, right? So in the heterogeneous RNA, a cap is added. A cap is added at the 5' end. The cap is added at the 5' end. Okay. And what is this cap? This cap is 7-methyl guanosine. Okay. This cap is 7-methyl guanosine, right? So for mRNA processing, a cap will be added at the 5' end, that is the 7-methyl guanosine, right? And the process of tailing, a poly A tail is added to the 3' end, right? So to the 3' end, we are going to add a poly A tail, right? So there is a stretch of adenine that will be added to the 3' end, right? So here poly A tail is added. Okay. So why capping and tailing is required? Because in the cytoplasm, this particular mRNA that is composed of nucleotides can get degraded, right? So we need to conserve our mRNA, we need to stabilize it and increase its stability, right? So as a result, capping and tailing is undergone by the mRNA, right? It's a very essential process. Now let's see about splicing. Now eukaryotes, they have split genes. What does split genes mean? It means that the coding sequence in their genome is intervaried by non-coding sequences, right? So we say we have split genes, simply meaning that in our mRNA, that is hnRNA for now, right? hnRNA. In the hnRNA, there are two types of sequences that we can see. There are exons and then there are introns. Okay, so there are exons and introns in our gene. Exons are the coding regions. And these coding regions can code for amino acid for proteins. Introns, they are the non-coding regions. They are interfering in the coding region, non-coding regions. Okay. So as a result, we say our genome is split gene because it is intervened by these non-coding sequences, right? So for an mRNA to be able to code for proteins, it should first undergo the process of splicing, wherein introns are removed. Okay, and the remaining exons, they are joined to each other. Now to bacteria or your prokaryotes, they have split genes? No. In case of prokaryotes, there is no split genes that are present. In their genome, only coding sequences are present, right? So there is no required of splicing in case of prokaryotes. So once this processing is done, our mRNA is ready. Our mRNA is ready to be transported to the cytoplasm. Now the question arises, if introns are not necessary for coding, what is the function of introns? Why are they still present in our genome, right? So some scientists have referred to introns or the non-coding sequences as junk DNA, but in no way are they junk DNA. They do not code for proteins, but they may have regulatory functions and they have very important evolutionary perspective. Using these sequences, we can trace who our ancestors were, right? So we can trace our lineage. Okay, so this we are completely finished with transcription. So what we have studied, we have studied transcription and RNA processing, right? So next we'll do translation. Okay, translation. So in translation, what is happening? The mRNA that was formed will now code for proteins or a polypeptide chain. But what we need to know is mRNA is composed of nucleotides, whereas proteins are formed of amino acids, right? So both of their language are different. Both of their language are different. So we need a sort of intermediate molecule, or what we say an adapter molecule, that can both understand the nucleotide language as well as the amino acid language. So this adapter is your tRNA. tRNA is an adapter in the process of translation, right? So tRNA, if we closely look at the structure of tRNA, it has a clover leaf model. It has a clover leaf structure, or we can also call it as inverted L. Okay. So in the tRNA, we have a 5' region and we have the 3' region. And we see that the 3' region is more elongated than the 5' region, right? Now it is to the 3' region that a particular amino acid binds to the tRNA, right? It is at the 3' end that a new amino acid binds to the tRNA. And we see that it has certain double-stranded regions. The double-stranded regions are known as stem. Right. The double-stranded regions in tRNA are known as stem. And then we have certain loops, right? This loop right here is the anticodon loop. Anticodon loop. And then we have T and D loops. Okay. Now there is an anticodon loop, and in this anticodon loop there are 3 codons present. There are 3 codons present here. Okay. There are 3 codons present in the anticodon loop, right? So let's also look at the structure of mRNA. In mRNA, there will be a sequence that is going to code for proteins, right? There's going to be a sequence that is going to code for proteins. Beyond this sequence, we have certain regions that are going to be untranslated, that will not code for proteins. These are known as UTRs. Five' UTRs, and then we have 3' UTRs, right? Okay. So for translation, we require mRNA, we need tRNA that is going to adapt as the adapter molecule, and then we require ribozymes or ribosomes. We require ribosomes. Now ribosomes are helping in the protein synthesis. They are protein manufacturing machinery, right? And ribosomes, they are made up of 2 subunits, a larger subunit and a smaller subunit, right? And the type of ribosomes present in prokaryotes and eukaryotes are different. So in case of prokaryotes, we have 70S ribosomes, whereas in eukaryotes, we have 80S ribosomes. Okay. S refers to the sedimentation factor, that is Swedberg. Okay, Swedberg. Okay. So this 80S is made up of 40 and 60S. Okay. Whereas the 70S is made of 50 and 30S. So we see that the ribosomes are different in case of prokaryotes and eukaryotes. And these ribosomes, they are formed from proteins and ribosomal RNA, right? Proteins and ribosomal RNA. Right. So we have learned about some of the ribosomal RNA like 28S RNA, then we have 5' sRNA, right, and 5S RNA, right. So in case of prokaryotes, we have 16S RNA. And this 16S RNA has been used in for classification, right? Has been used for classification purpose. Okay. So ribosomes have a small subunit and a large subunit. So when does the process of translation begin? It begins when an mRNA is encountered by the small subunit. We have the mRNA here, right? And this mRNA, when it encounters the small subunit, then the process of translation starts. Usually, the ribosomes, both of the subunits remain dissociated in the cytoplasm. So, small subunit is first encountered by the mRNA, and when it encounters mRNA, that brings the larger subunit in association to the small subunit. Now, we have the complete ribosome and the mRNA. Right? So in mRNA, the codon that is going to start the process, or we can say the one that is going to start the process of translation, or the start codon, is always AUG. Right. So the first codon that is going to start the formation of protein will always be AUG. And this AUG sequence is going to be present in the mRNA, right? And we also have stop codons. We also have stop codons. Out of the 64, 3 are stop codons. We have UUU, UAG, and UGA. Right? All of these are stop codons. So let's come back to this question. Okay, so the first codon will always be AUG. Right, and AUG is going to code for methionine. AUG codes for the protein methionine. Right. So in both prokaryotes and eukaryotes, the first amino acid to any protein will always be methionine. Right. The only difference is in case of prokaryotes, it's formulated methionine. It's formulated methionine. That's the only difference. Right. Okay. So now, for a particular codon, we require a particular amino acid. Then this amino acid is brought about by the tRNA. This is brought about by the tRNA. Okay. Now in ribosomes, we find 3 particular regions. I'll just show a separate diagram. So there are three regions within the ribosome. Okay, we have the A site, we have the P site, and then we have the E site. Okay, so A site is where tRNAs are going to bring your amino acid, right? So your tRNA, I told you, can bind with an amino acid, right? Can bind with an amino acid that confers to the codon, right? Depending on the codon that they have, they will bring a specific amino acid, right? So when tRNA binds with amino acid, in this particular state, it is said to be charged. It is said to be charged. Okay. So at the A site, always a charged tRNA will come and bind. So here we have a tRNA that is charged. It will come and bind to the A site. Okay. So that is the case usually. But for the first tRNA, that is the initiator tRNA, it will not come at the A site. Rather, it will come at the P site. This is an exception and occurs only for the first particular tRNA, right? So the first tRNA is going to come and bind to the P site rather than the A site. Okay. And the first one, initiator tRNA, will always have which? It will always have methionine, right? It will always have methionine. It will bind with AUG in the mRNA, right? So it binds with the P site. Now, there would be next charged tRNA that is coming along with an amino acid in respect to the whatever codon that we have here. Okay, so here we have another charged tRNA that has another amino acid, right? So when two charged tRNAs, they are very close with each other, they are in very close association with each other, that helps in formation of a bond between these two amino acid, right? So when two charged tRNAs are in very close proximity with each other, that energetically favors the formation of a peptide bond. Peptide bond. Now we know that in proteins, amino acids are joined to each other with the help of a peptide bond. Right? So when two charged tRNAs are very close to each other, their amino acids are going to form the peptide bond, right? Now when this peptide bond is formed, what will happen? This amino acid will get transferred to the chain of this existing tRNA, right? So here I have a new amino acid, right? This comes and binds to the P site. Okay. So can I say now this tRNA is charged, right? Okay. Okay. So once the peptide formation is done, the peptide chain will be transferred to the A site. It will be charged to the A site. Okay. It will be changed to the A site. Now the P site tRNA is empty. It is empty. So the empty tRNA will move to the exit site. It will move to the exit site. Right. So this empty charged tRNA will now move to the exit site from where it is going to get exited, or it will move out of the ribosome. Okay. Next, what will happen? The charged tRNA that has all these different chains of amino acid will now move to the P site, right? Will move to the P site. And again, a new charged tRNA will come that will bring a new amino acid, right? And peptide formation always occurs at the P site, okay? Always occurs at the P site. And the empty tRNA will move to the exit site, right? Okay. So our ribosome has a property of translocation. What is translocation? For example, now once we have the AUG tRNA here, we have a tRNA here, right? So all the tRNAs are in position. After they bring the amino acid, this ribosome can move by one codon length, right? So that particular region can be made empty again, so that new tRNAs can come and bind to the A side, right? So that the whole ribosome will move by 1 codon length, right? When it moves by 1 codon length, the A side becomes empty, right? The P side moves to the E side. So translocation is there, right? And another property of ribosomes is they can form peptide bonds. They can form peptide bonds, okay? So ribosomes can translocate and they can form peptide bonds. So how is termination occurring in ribosomes? Okay, so now if in place of a charged tRNA in the A side, a tRNA comes that is bounded, a tRNA comes, but here the codon is a stop codon. If in the A side, the codon that is red is a stop codon. There are 3 stop codons, right? So if in the A side a stop codon comes, this stop codon is not recognized by any of the tRNAs, right? It is not recognized by any of the tRNAs. As a result, what will happen? No tRNA will come and bind to the stop codon, and as a result, the process of translation will stop. And the polypeptide chain that is formed will be released. It will be released. Okay. And proteins, we know they have an N terminal and a C terminal. Okay. So this protein will always have an N terminal and a C terminal. After this protein is formed, it is going to undergo folding. It will be properly folded, right? It will be properly folded with the help of chaperones, and then it will form a functional protein. After only a protein is folded properly will it be functioning properly, right? So we require chaperones for that process. Okay. Chaperones. Okay. Next, let's talk about regulation of gene expression. So we now know how a genotype or a gene expresses itself in the form of proteins, right? But we need to regulate this protein because the proteins that are formed in our body are going to perform functions. So it's very important that a correct protein be made. So this whole process is regulated at a number of steps. In case of prokaryotes, because in them the process of translation and transcription both are coupled, so much of the regulation happens at the transcription level. Okay, it is at the transcription level that much of the regulation is happening. But in case of prokaryotes, there are various levels. Sorry. In case of eukaryotes, there are various levels at which regulation can be done. It can be done at the level of transcription, it can be done at the level of translation, it can be done at the level of RNA processing, right? It can be done at the level of post-translational changes. So there are various stages in which eukaryotes can regulate their gene expression. Okay. But for prokaryotes, much of the regulation is done at the level of transcription. Now we have to study about operons. Operons. Now, in case of prokaryotes, the transcription unit that we have studied is a little bit different. Prokaryotes, they are polycistronic, whereas eukaryotes, they are monocistronic, right? They are polycistronic while eukaryotes are monocistronic. What is meant by polycistronic? We'll try to understand. Okay. Poly means many, right? And cistron simply refers to genes. Prokaryotes are polycistronic means on transcription of a particular unit, they are able to transcribe multiple genes. They are able to transcribe multiple genes. As a result, just by the transcription of a particular unit, they get multiple proteins, right? They get multiple proteins. Whereas in our case, in case of eukaryotes, each transcription unit has one structural gene. As a result, through the process of transcription and translation, we get just one polypeptide from one gene, right? So eukaryotes, they are monocistronic. Monocistronic. So when we see the transcription unit of prokaryotes, what we are able to see is, again we have a promoter sequence, right? We have a promoter sequence similar to your eukaryotes. We have a promoter sequence, right? But now before the structural gene, we have an operator. We have an operator. And then we have the structural genes. And then we finally have the terminator. Okay. So this is the transcription unit of a prokaryote. So what is different in a transcription unit of a prokaryote and a eukaryote? First visible difference is the presence of an operator region. An operator region is not present in case of eukaryotes, while in prokaryotes it is there. Another difference in respect to structural genes. What I told you, in case of eukaryotes, we just have one structural gene, right? While in case of prokaryotes, we have multiple genes here. There can be gene A, gene B, and gene C. Okay? So what we observe is all of these three genes, they have a common promoter and they have a common terminator. So all of these genes are regulated as a unit. They are regulated as a unit, right? Whereas in eukaryotes, each gene has one promoter and one terminator, right? So in prokaryotes, why is there such a particular sequence or organization? It is because in case of prokaryotes like bacteria, they are unicellular forms and they have to constantly analyze their environment, right? And depending on the environment, they have to change their gene expression, right? And we know, of course, in a metabolic pathway, there is no one-step reactions. There are rather many small-step reactions, right? And in all these particular pathway, there are multiple enzymes involved. In a particular pathway, there are multiple enzymes involved, right? So all the genes that are required for this pathway, they are present in a particular transcription unit, right? So what happens? Just when you transcribe this particular unit, you get all the enzymes for a pathway, right? So it's much more faster. It's much more faster than in case of eukaryotes. Okay. So this whole unit is known as the operator. From the promoter to the terminator is known as the operator. Okay. Is known as your operon. Operon. Okay. Now besides having an operator, operon also has a repressor. It also has a repressor. The operator in an operon will act like a switch. This is going to act like a switch. Okay. So in operon we have repressor, promoter, operator, structural genes and terminator. We have all these sequences. A repressor gene will code for a repressor. Right. It's going to code for a repressor. Okay. Now this repressor, what will happen? This repressor can go and bind with the operator. Okay, this repressor can go and bind with the operator. So now, if repressor is present here, what is it doing? It is stopping the promoter and the structural region, right? It is present in between these regions. So to the promoter, what comes and binds? RNA polymerase comes and binds, right? So if repressor is here, it will stall the RNA polymerase. The RNA polymerase will not go ahead and transcribe the genes, right? So presence of a repressor is going to repress the process of transcription. The RNA polymerase will not be able to move forward. It will be stalled at the promoter region, right? So what would happen if there is no repressor? If there is no repressor, the RNA polymerase is free. The RNA polymerase is free, right? So now it can go ahead and transcribe all the genes that are involved in the pathway. Okay. So let's quickly do an example of this. Let's take the example of lac operon. Lac operon. Okay. Lac operon is an inducible operon. It is an inducible operon because this operon can be induced. We'll understand the word what is inducible operon. Okay. So if imagine this is a lac operon. A lac operon will have the genes that are required to metabolize the sugar lactose, right? It will have all the genes that are required to metabolize lactose. Now, like glucose provides us with energy, in bacteria they can either use glucose or lactose, whichever is present in their media, right? If glucose is not present in their media and they have lactose, they'll be using lactose for energy production. Okay, so we have genes that help to break down lactose, right, and to use it as an energy source. Okay, so in case of lac operon, we have 3 genes. We have Y, A and Z gene, right? Y, A and Z gene. Gene Z is going to code for beta galactosidase. Beta galactosidase. Okay? Beta galactosidase is going to break down your lactose molecule. It is going to break down your lactose molecule into galactose and glucose. Okay? This one is responsible for breaking down of lactose. Okay. Okay. Then we have gene Y and A. Gene Y and A, they help basically to increase the permeability of the cell to lactose so that the lactose can be uptaken by the bacterial cell, right? And there is a transcytylation process that would occur, right? So these 2 genes help in that process. Okay. So when lac is present in the medium, when lactose is present in the medium, my bacteria has to take lactose and utilize it, right? So this operon should be functional then. But when lactose is absent, this operon should be switched off, right? So when lactose is not present in the medium, what is happening? The repressor goes and binds to the operator and stops the lac operon, right? And stops the lac operon. Now when there is lactose present, what will happen? The lactose is going to act like an inducer. The lactose will come and bind with the repressor. Okay. Now the repressor is not free to go and bind with the switch. So what will happen? So here the operator region is free, right? The repressor is not bound to the operator region. So now the RNA polymerase will come and bind to the promoter and transcribe all the genes required in the lactose pathway. So that is the why we say lac operon is an inducible operon. Why? Because this operon is normally switched off. It will only be switched on when lactose is present, right? Then only it will be switched on. So we say this type of operon is an inducible operon. It can be switched on whenever there is requirement. Okay? Similarly, we can have repressible operons. Now repressible operons are those that are always switched on but are switched off at certain times. Okay, those are repressible operons. Okay, so we can have inducible operons like that in case of lactose, or we can have repressible operons like in case of tryptophan. Okay, tryptophan operon is an example of repressible operon. Okay. So we have also studied about operons. So in the next session, we'll continue.